My name is Stephanie, and my topic today is animal testing. My main claim is that the outcomes of animal testing are not beneficial. Before I move on, I'm going to explain the two types of research that I talk about today. So there's applied research, which is used to find solutions to everyday problems, cure illness, and develop innovative technologies. And then pure research, which is exploratory in nature, is conducted without any practical end use in mind, and it's driven by gut instinct and curiosity. So my supporting claims are that animal testing is bad science. There are more effective and cheaper methods. There are more effective and cheaper methods than animal testing, and animal testing hurts and harms innocent animals. So back to my first supporting claim, which is animal testing is bad science. Modern research in recent years has proven that animals rarely serve as good models for the human body. People that do accept animal experimentation do so only because they believe it to be necessary for medical progress, when the reality is that most of animal experiments do not contribute to improving human health, and the role that animal experimentation plays in most medical advances is questionable. Humans and animals have very different physiology. Our closest relative is the chimpanzee, which we share 98% of our genes with. But in 2011, the Institute of Medicine concluded that there was no current need for chimpanzees in biomedical research. The NIH responded by retiring 90% of its chimpanzees. So if our closest relative is not useful for human research, why should other species be? Relying on animal testing is like relying on blind faith. Many drugs that pass animal tests aren't safe in humans. The Food and Drug Administration has said, currently 9 out of 10 experimental drugs fail in clinical studies because we cannot accurately predict how they will behave in people based on laboratory and animal studies. Even scientists are aware of flaws of using animals as models of human disease. There are many examples where reliance on animal models have proven to be misleading and detrimental to human health. In the 1950s, there was a study on rabbits for a medicine that was deemed safe. The medicine was to help with morning sickness on pregnant women. A year later, it was found out that the drug caused 10,000 birth defects. Drugs that are deemed unsafe for animals oftentimes end up being safe for humans also. One example is a very commonly used penicillin. It was ineffective in rabbits and guinea pigs, but it later proved to be safe and effective for treating bacterial infections in humans. Another example is aspirin, which showed negative effects on animals, but now hundreds of thousands of people use it in their daily lives. There has been a cure for cancer for many years in mice. According to the former National Cancer Institute director, Dr. Richard Klausner, he said, "We have cured mice of cancer for decades, and it simply didn't work in humans." So research on animal testing wastes time and is ineffective, leading me to my second claim, which is there are more effective and cheaper methods than animal testing. So animal testing is done for many types of research. The research ranges from applied to peer research. The testing and experiments done are extremely expensive. Most of this research is funded by the federal government. According to an analysis of federal government, the National Institute of Health spends between 12 and 14.5 billion on animal testing every year. We could be saving taxpayers money by using alternative methods of research that are cheaper and more effective. Some of these methods are in vitro testing. So Harvard Wyss Institute has created organ-on-chips that have human cells that imitate the structure and function of our organ systems. The chips can be used in place of animals for researching disease, medications, and toxicity on human body. Advances in technology have also let researchers create in silico, more commonly called computer modeling, that stimulates human biology and accurately predicts the ways that new drugs will react in the human body. These methods of research are less costly, use our human bodies to more accurately calculate research, and most importantly, don't harm animals, which leads me to my last and final supporting claim that animal testing harms and kills innocent animals. The Animal Welfare Act was signed into law in 1966. It's the only federal law in the U.S. that regulates animal testing. The AWA excludes roughly 95% of the animals tested upon, such as rats, mice, birds, fish, and reptiles. It provides only minimal protection for the rest and has no authoritative power over labs because they're not even required to report non-AWA protected animals. Some of the cruelties that go on in labs are animals are being shocked, poisoned, isolated, starved, drowned, addicted to drugs, and brain damaged. One of the worst cases of cruelty that goes on in labs are the vivisection, and it's where animals' limbs are cut off while still alive. Over 100 million animals each year suffer and are murdered at the hands of cosmetic, chemical, drug, and food companies. Medical research labs and university labs, after animals are severely hurt and experimented on, if they are not useful, they just get killed off or left in their cages to die from their severe wounds or traumatizing experiences. So to sum up my speech to you all, with new technology and more effective alternatives, animal testing is outdated and unnecessary. Using alternative methods, we can reduce government spending and also put a stop to the torture and harm to animals.
Right, well, the proposition is labeled clearly, and I thought that there was a good preview. The controversy seems to be about whether or not there's necessity on this, which means that the third point is really not an issue of fact, and it doesn't fit to the claim that you're presenting. You've got an explanation of applied research and peer research, and then you never talk about whether or not one works and one doesn't, or whether or not we've learned anything from either type of research. So I'm not sure why you bothered to define those two types, since they never come up again as part of the discussion of your argument. You signpost the first point very clearly. And I think you did a nice job outlining the challenges, some of the comparisons that are being made. I like the idea that if chimpanzees can be excluded and the NIH is essentially said that they can't be used at all, then the argument seems to follow that if they are the closest to humans, then why are we allowing any other animals to be utilized? I think that's an interesting point. However, I do think that there are probably comparisons that are being made, some of it in that peer research and applied research stuff that might be leading to information. For later on, the subject comes up, for example, about curing cancer in mice, and the first question that popped into my mind is, why is it that the cures in mice don't work in human beings? That seems like it would be something that you would spend years studying so that you could find answers that would benefit human beings, but there's not really any explanation about what has resulted from those particular issues. If there's no knowledge that's been gleaned from that, that would be problematic. Now, the best argument that you have when it comes to benefit of human beings is the evidence that says that 9 out of 10 these products get, or 9 out of 10 of these chemicals get put on the market, and they end up not working even after they've done the animal research because the animal models are inaccurate. I think that that's pretty helpful, although there are some questions about safety issues. The subject that you're talking about from the 1950s, the medication is thalidomide, you didn't name it by name, but that's what it is not really, you don't tell us whether or not there were any human tests on that before it was applicable, and in fact the very experience that you're talking about is one of the reasons that the FDA requirements went into effect in 1962 for efficacy rules as well as for safety rules. So the question then becomes, would a different type of animal testing, or for instance the second stage of human testing, have eliminated that? That I'm not sure shows that the animal research is useless. It simply means that it was useless in this particular case. I thought you did a nice job citing a variety of evidence on points. I don't think that the alternatives are as well developed as they needed to be. It sounds very hypothetical on a couple of these points. Now, if you can demonstrate, for instance, that we've had these kinds of breakthroughs, because that would be the thing that the opponent's going to be talking about, is all of the medical advances that have been a result of animal research. And the first question is, okay, if we don't need animal research and we can do these other kinds of things, have you got data that shows that these other things have produced these kinds of medical breakthroughs? That's what I think needs to follow through on. All right, like I said, the third point really feels to me like it's all a value argument, and it's not really related to the claim of fact that you're dealing with. All right, thank you.